Welcome back to the second part of the November iReceptor seminar series, now with Jarrod Shilts from the Sanger Institute, and he's going to talk about receptor wiring networks in the immune system.
Great. Yeah, thank you very much for that, Victor. Let me just share screen on this. Well, hopefully that's looking all right. So anyway, thanks everyone for coming, particularly for those of you in America spending your holiday morning listening to me, learning about immunology. Very good. Today we're going to be doing a slightly different topic than I think some of the more recent seminars, where we're going to be looking a bit more broadly at not just, say, some of the variable receptors in the immune system, but what are the repertoire of immune receptors? How do they function? How do they cooperate together? So hopefully it should be a nice different perspective in some ways I could introduce. And so kind of as a motivation behind this, looking at the initial problem, if we imagine this kind of textbook image of two immune cells interacting, like your T cell in blue and some antigen presenting cell in red. Normally, as this would be presented right in your textbook, is you'd have, just as it's shown here, a few examples of a few receptors, like say the TCR or some of these other well-known activating or inhibiting molecules on the surface that then can bind, mediate the communication between those cells. And it's kind of all packaged up as a nice, neat story. But of course, if you actually look on a T cell surface, there's a lot more receptors and surface proteins on their surface than are encoded here. Just kind of as a little fun illustration of this, there's a current catalog of, on an average immune cell, several hundred of these that'll be on any individual cell. Then you say have 100 more on the other cell that's interacting with. And so what initially looked like quite a simple picture, when I first illustrated it, is actually hiding quite a lot of complexity underneath between what are all these hundreds of other receptors doing that we're spending much less time studying, understanding what their functional roles are. And that's kind of been the goal of this project, using the immune system to try and study what are the roles of all these different surface proteins, how do they interact with each other, and then how do they actually mediate connections and communications between immune cells, which I think the immune system is really a fascinating case study in this, because most of the populations there are highly migratory. They're going to be moving around quite dynamically, encountering another cell, interacting, and then parting off to communicate with some other cell. And so those hundreds of other receptor pathways and surface molecules, quite clearly would have some kind of functional role in that process in maintaining a cohesiveness for a cell system that's all, single distributed units across the body. Just kind of as one nice little illustration of this, because I also like videos of give you intuitions. If we just take even just a single cell pair of two immune cells, in this case, our classic T cells and dendritic cells interacting, even in the absence of specific antigen signals that we're used to, being associated with classical immune responses, as you can see in the video, these cells are still forming connections. They'll bond together on the order of minutes, and there's clearly something happening there, but it's a much less studied process than your famous, say, TCR signaling pathways. And so hopefully that kind of sets up some of the challenges and the aspirations for this. Is if you have some cell types in the immune system, showing their surface proteomes here with all these different proteins that you can detect on their surface. The goal is, can we create these, what I call these wiring diagrams or wiring maps of what is connected with what, how strongly, and then if you do this across multiple immune cell types, can we actually get a sense of just how the immune system is held together through some of these linkages. In terms of how we actually do this experimentally in the lab, I'll kind of give a brief conceptual overview, since some of the methods I used are, I think, a bit different than some of the past ones in this seminar series have been discussing. So it's a assay that we have where, say, for these two interacting cell types of interest that you want to study and figure out what are all the ways that their surface proteins can interact between them. We isolate the extracellular regions of the surface proteins on each cell surface, encode those in synthetic DNA constructs, then express those in human cell lines as these recombinant fusion proteins with kind of different linkers and stabilization regions. So they somewhat resemble the structure that you'd have embedded in a membrane, at least in terms of their binding functions and those kind of properties. And then as the last step, we can then multimerize those and lay one onto a solid surface, which we'll call a bait in some of the later slides that I'll show. The other one also multimerized, but in this case around a reporter construct, and I'll call that the prey. You can imagine a big plate or array of these baits with a different protein in each well. You can then wash over these preys. Wherever the two proteins interact, those will stick, give you a signal, and then you can use that at quite high throughput to deconvolve what's kind of the global receptor interaction network between two cells. So kind of illustrating this, you take cell A, cell B, purify all their proteins, and you can imagine, this is a small example, the actual things involved here involve, 1,000 384 well plates, but just as one example, you take a small subset here, and you can see these little blue dots showing you what's actually interacting between those two cell types to, again, try and get a systematic view of what's happening here. For this project, again, our ambition was to try and do this as comprehensively as we could, just to see, since no one had previously, to our knowledge, looked at a systematic survey of all the receptors, what they're binding to. It's been mostly hypothesis-driven studies. We wanted to say, okay, just do every measurement we possibly can and see what discoveries come out of that. So to figure out all the surface proteins we had on immune cells, we combined empirical data sources from proteomics, where they isolated cells, measured what's on their surface, and then plus good community curated resources where there's also data on what's on their surface. And from that, combining together about 44 different cell types and cell states of different human immune cells, particularly the leukocytes in blood, and then identifying 630 unique cell surface proteins that we're able to express and isolate through our expression method. That's, of course, quite a large number, so I'll kind of talk through how we did some of this to get our full interaction map. Within those 630 proteins, they have very diverse structures, which is actually something a little bit unusual about some of the approaches that we took. We tailored our expression constructs so we could design ones that encompass the true diversity that you would see on immune cells, where some things are, say, heterodimers, as complexes, others might be, type 1 or type 2. So without going into the details, we've designed ways that we can engineer that with our fusion proteins that we build together. And then high throughput systems that allow us to essentially produce and purify these at quite high scales. So you can make, hundreds of these proteins within the matter of a few days to a week. Allowing us to, I think, for the first time, do these really kind of ambitious experiments to answer for the field, just what does this look like? What does it look like when you measure all these receptor interactions against each other, to build, I think, a really important missing resource for immunology. The final matrix, if you squint hard, you may see some of these dots here, but this is in fact close to 500,000 measurements that we took, pairing together every possible combination of those proteins that we isolated out. So again, to not be predisposed by a certain hypothesis or thing that we're going down, just do every measurement and then see what the data tells us is actually happening in the immune system for all these interactions. For every novel interaction that came out of this, we then had several layers of validation, say repeating it in different formats, doing cell-based assays, doing assays like SPR to get a more quantitative sense of its binding. And through all this kind of meticulous process, we were then able to assemble this, which is our kind of final schematic depiction of the interactome of human receptors. You can see through all the red colored edges how just through the study alone, we've been able to expand what was known about the human immune system by about 20%, just to get a sense of, how much was previously unknown, how much we're able to fill in to actually get this systematic picture of what happens in the immune system. I'll just go through a couple of case studies of why this is quite interesting, particularly for the cell biologists in the audience who, realize every single one of these links could be quite a fascinating story in themselves. It's like one of the discoveries we made through this process involved a receptor called VISTA, which is a checkpoint inhibitor for T cells. And I think quite interestingly, we found these non-canonical MHC proteins, HLA E and F, that have quite interesting regulation on their cell surface when they're presented on the cell surface, I should say, and are also known immunomodulatory ligands for, say, innate immunity through natural killer cell interactions. And so it, I think, fills in a little piece of the puzzle there in terms of how did this mechanism that balances innate immunity can also be adapted by T cells to maintain their quiescence. Just as one example out of this, Another very neat case study, which I also quite like because an independent group at Genentech independently discovered this around the same time and validated it, an interaction involving this inhibitory KIR receptor and a ligand associated with different or certain types of tumors where it may be overexpressed, inhibit the NK cells. This is now already under investigation to try and translate this and see, can we use this in immunotherapy contexts? And so the ways that when you do this, you discover links like, say, this KIR and PVR link. There was no previously known interaction between a KIR and a protein like PVR in that family. And so just showing the power of these systematic approaches and some of the novel biology that comes out of it. In addition to each of those kind of biological vignettes, I think there's a lot you can do at the systems level analyzing this data. And what was really neat about this project is when we did the surface plasmon resonance on every interaction, it gave us not just the network of what's connected, but a fully quantitative network where we know how strong those connections are. Again, to my knowledge, this is, I think, a somewhat unique resource to actually have that measured so comprehensively across, more than 100 different receptors and their interactions. And so with a quantitative sense or, quantitative resource to give us a baseline for what happens in the immune system, it really opens up a whole range of interesting analyses that you can start to do, and not just analyses, but mathematical models, as I'll illustrate in a second. This can range from just very basic questions, like if you take two interacting cell types, here I did the example of a professional antigen presenting cell, which could either be a B cell or a dendritic cell. We know they interact with T cells. That's, of course, quite important for their activation. But curiously enough, they vary in the affinity of that interaction, how strong the receptors bind on to the receptor on the T cell. And just another interesting analysis that came out of this, if you look at immune cells that are in the resting state, compare those to activated immune cells of those same populations, there's this quite broad, I think what we call an affinity switch, where there's a bias towards some of these lower affinity interactions, which I should say are the higher numbers, because this is measured in terms of KD, but some of the lower affinity interactions at a resting state as immune cells are surveilling and moving throughout the body. And then that quite dramatically, they downregulate those receptors and replace them with high affinity receptors upon activation. Just again, as an example of the kind of things that this opens up once you have this resource. And then I mentioned modeling as well. If we measure these molecular components in this level of detail, and we did our job properly of doing this comprehensively, it should follow logically that you can actually model from basic physical principles how immune cells associate. Just to give an intuition of this, it's fairly intuitive that if more cells have more connections between them, that means they probably are more likely to interact or have long-lasting interactions, which that network I showed you before it sort of gives us the data to address. Then, of course, receptors that are more abundant can also be better at mediating these long-lasting associations between cells, which you can get from a wealth of proteomics and other data sets in the field. And then the strength of the interaction, of course. So weaker interactions will be less able to mediate cells to interact, and then stronger interactions, which our affinity measurements give. And then just through basic kind of physical formulas like mass action laws, you can combine those different numbers I mentioned together and actually produce, just through the data that we measured from all these different sources, combined together using these kind of physical formula, and produce predictions that actually agree with some of the things that you can observe under a microscope. Like in this example, where we took a published data set where they measured the rate at which different immune cell populations physically associate with one another. And mathematically, we can reconstruct the rates to at least a fairly good accuracy just through this, reductionistic first principles model. I think just showing the power of some of these approaches and just what becomes possible when you take a systematic perspective on some of these things. Then, kind of as a last chapter on this, more opportunities for integrating data sources for, again, some of these more global views. So there's been, particularly over the last few years, a wealth of studies taking different tissues in the human body, measuring the immune cells in them, what they express. And we realized that created a really nice opportunity where we could take those resources, integrate them with our interaction maps, and then create kind of this website down at the bottom, an interactive resource for the community where you can take tissues throughout the human body look at their immune cells, look at the receptor connections between them, what cells are talking to each other more than others, and enable a range of analyses like that to, again, this is at least a global overview of that larger data set to really provide this resource and all the things that you can do with it. In the time that I have, which is not too long, I'll just highlight one example where within these maps, you can actually convert them into graphs. So, like the sense of graph theory, where you can look at the connections between cells and how strong those are. And when you do an analysis like that, you can actually find, so to speak, these hubs within cell-to-cell interaction networks in different tissues of the body. In this case, we had quite an interesting result where myeloid lineage cells seemed, across actually multiple tissue types in the body, to be much more centrally connected in these cellular signaling networks mediated by these receptor connections. So this is just one example out of many, but I think it hopefully illustrates what this enables then once you have kind of a firm foundation that's all systematically gathered and avoid some of the biases of some of the previous, say, databases or other sources that were available. And then for the last section of this, I think one of the more interesting parts, we of course discovered many interactions during this project, and to actually understand what they might do on immune cells, we also came up with some technology, sort of development and optimization to look at those questions. And the way I would frame this is for each of those little red links I showed earlier where we discovered something, it's really kind of like, or finding what it does is really like finding a needle in a haystack. There's all kinds of functions that could be mediating the body, like resistance against cancer, extravasation from blood vessels, cell division, activation. And by expression data alone, you don't even really know what cell type it would be most likely affecting. Right? There's just so many possibilities there for functional phenotypes. And usually we just come up with a hypothesis, test it. Maybe you get lucky and it works, maybe it doesn't, but you're kind of biased in that sense. So we wanted to create a method that allows us to more globally survey what functions these novel receptor interactions are having, where we could isolate out primary human leukocytes, stimulate them with purified recombinant proteins encoding the binding partners we discovered in our screen, and then use this really nice multiplex microscopy method that was invented by our collaborators at ETH Zurich and Bernd Schneider's lab that allow you kind of in this same kind of analysis pipeline to look at cell activation states. So things like, our T cells being suppressed or stimulated to proliferate, in the actual interactions themselves to measure those at a direct level. So I'll just go through this relatively quickly since this was recently published and you can look at some of the details there in the time that we have. But we could then take for this panel of different receptors that we found novel interactions for, stimulate them across different lymphocyte sets in this particular example, and find new immunomodulators, like this example of SLTRIC4, which is quite interesting, that could quite potently activate NK cells, to some extent B cells when they're in a resting background, not having been activated before, as well as some of these other phenotypes that you can extract out that you wouldn't have otherwise guessed, right? There was no prior hypothesis leading us to that, but these methods can point you towards. And similarly, quite interesting if you look at just the rates of cell associations themselves. So by infusing these recombinant proteins, you can block those links on short time scales. So you're just interfering with the interactions without hopefully inducing any signaling. And you get these quite fascinating patterns of changes. There's different cell pairs color-coded along the bottom. But you can see, you can essentially read out from these kind of assays how each receptor contributes to global cellular connectivity in the system. Of course, we repeated that across our full set to really just characterize, particularly for some of these lesser studied interactions, say like what does a B cell and a T cell when they interact, less studied than say a T cell and a dendritic cell, of course. And so characterize what mediates those interactions, what can we functionally link them to through our assays and other techniques available to again try and answer this really important question in a more hypothesis-free way. So just to summarize everything, the minute that I have left, starting off with deciphering receptor wiring diagrams, just figure out what is the sort of global landscape of connections between two cells or even multiple cells across the whole immune system, using functional assays to inform us what those new interactions we discovered do, what consequences they have on the cells. And then these really nice data integration methods that are advancing by leaps and bounds every day and becoming more sophisticated now we can plug in our better data sets into them and really get some insights into just how the immune system is held together and coordinated between its constituent cell types. Yeah, so anyway, just a last note about where we can go from here. There's each of these new interaction links, right, could be a fascinating story on its own right. There's a lot of systems biology, as I gave some examples before on, and at least I'm personally quite interested in the cellular engineering aspects of this. And for those of you who wanted to know more about this, I think there'll be time for questions later on, and happy to answer those. And so kind of globally, my vision for this, and like the analogy I drew at the start, is having this kind of be a wiring diagram for the immune system, where we take, in a relatively standardized way, all the components, measure those components, and assemble them together through this rational framework, so we can predict at the systems level just how the immune system functions and what these receptors are doing, sort of beyond some of the more limited narrow views, but actually trying to do this at a system-wide level. Cool. So thank you very much for listening to that, and especially to everyone who helped put this quite large initiative together, particularly Gavin Wright, who's my supervisor during my PhD at the Sanger Institute, our collaborators at Zurich who are doing the microscopy assays, and then everyone else listed here for their help on modeling, analysis, doing the experiments, and everything to make this project possible. So with that, I think we have 10 minutes for questions, and happy to answer whatever is on your mind.
Beautiful data. On one of the first slides, you showed this matrix, this interaction matrix, yep, which was quite white, color white. So
yes,
quite
yes,
sparse, maybe. Could you comment on that? And then later on, you showed a network plot. Can you comment maybe on the degree distribution?
Ooh, yes. No, it's a great question.
Like
Let me just bring up the visual as well so folks can follow along with the slide that you were mentioning. So is this one, right, Victor?
Yeah.
Yeah, so this is an absolutely excellent point, and just in the time I couldn't elaborate this on as much, but if you compare this to any other interactome, this is remarkably sparse, right? It's 99.9% white space, and that makes the technique development quite challenging because you have to have incredibly stringent false positive thresholds to avoid, seeing interactions. But I think it also matches just our prior known intuition with a lot of these receptors, where they might just have a few binding partners, just any example you can name off the top of your head, CD28, CTLA4, rarely is there more than 3 or 4. And unlike intercellular proteins, you have this remarkable sparsity to the network. Now for your second part, going to the actual diagram themselves, I also love that part of your question because in the paper, we talk a little bit about this because to my understanding, this might be one of the first systematic receptor wiring diagrams of any system, not just immunology, but anywhere. So there's all kinds of things that you can glean just about how receptors are connected in human cells. Just one example that we highlight in the paper is if you see this big interconnected network in the center, which is one of the only ones where the different receptor nodes along the network are actually all fully interconnected, not just these more sparser kind of branches on the side. It's actually somewhat enriched for these adhesive receptors. And so there's some interesting kind of thoughts you could have in terms of why the signaling pathways along the periphery that have more directed function tend to be more sparse, relatively speaking, and these adhesive pathways have more interconnections between them and what that means. But we only kind of scratched the surface in our paper of that. So absolutely, I would encourage anyone who's curious on that, go into our networks, go into the data. And I think there's a lot more you can extract out just about networks in general and then networks in the immune system.
Great. But then we have 2 questions by Anastas and then Katharina.
Maybe the lady first.
You raise your hand first.
Please, someone go. Yeah.
With respect to the false positives and sparsity of this matrix, this seems quite realistic and relevant. And I admire this work. I really find it fantastic. Thank you. But I have one particular question. What were the conditions for the binding assays? when you are screening.
So the technical details, you mean like pH, that kind of thing? Is that what you mean by condition?
More like the composition of the buffer, because typically these binding assays in vitro are in very artificial molecular environment.
Oh, yes.
And especially with respect to the low protein content. And if you imagine that these interactions occur in plasma or in tissue fluid with all the proteins around and the variety of proteins around, how this would affect, because we know from the antibody binding that if you isolate antibodies from the serum, suddenly there is a abundance of autoreactivities.
It's a great question. Yes, so great question. We definitely thought quite a lot about that, because you're absolutely right. People have, when they've tried to do these, false positives are always the main thing on your mind. In our buffer, in addition to kind of the standard conditions of, having physiological pH, having osmolarity there, we also include enough serum protein to roughly mimic the concentrations you might see in human blood plasma. That said, I think another comment that I would like to make, just something I didn't get to show, is we also could benchmark our data set, because it was so large, against essentially all the knowledge the field of immunology has gathered over the past 30 years of discovering receptors. I think that, at least in my mind, was one of the most convincing pieces of data on this, is we could take, again, dozens of different labs across decades, across countries, what they discovered, and that we're able to recapitulate a majority of those in our data set. Of course, some of the disagreements were actually quite interesting to look into. Is that physiological, all of that? But from those, I think it confidence that the quality of the network we had was certainly kind of good enough that we could do things like the mathematical model, do things like the cell perturbation assays, confident the interactions we're claiming are actually real.
Okay, thank you. I'm glad you said that, because I was sure you had done something like that, because it looks very realistic. But don't forget that most of these other labs had done these binding experiments also probably in not very realistic environment. Anyway, it looks true. And now I would just like to
we have to keep the questions really short.
Just one small question.
Okay.
Sorry.
Yeah.
Victor. There are a lot of natural antibodies that might interact with these receptors. And back in the 90s, there was one fantastic idea about immunosomatics, that these natural antibodies could be actually pushing the buttons here and there, and their affinities are in the same range, actually. So just a hint, an idea that you could just in your spare time try to see if you put a lot of, let's say, IVIG or IgM isolated from healthy person, and see how this changes some of these interactions. Thank you.
Oh, very nice. Yeah, thank you for that.
So then we have Katharina and then Gur, and then I think we can round off.
Yeah. So I agree with everyone else that cool data.
Thank you.
I'm going to ask a little question. So how accurate is the method? So in which range can you measure? And do you think you would be able to detect differences between different alleles that interact? For example, I think here and HLA-F, where it's known that there are differences.
Yes. So this is a good point, particularly for those of you quite interested in the variable regions of receptors. So when we designed this, our goal was to make it sensitive enough that it could even detect the binding of quite very weak interactions. So when you talked about some of those alleles and their variation in affinity, I think, I'm not an expert in this, but most of the variations that I'm aware of would still be strong enough to be detected in our assay. Essentially, if it's 200 micromolar or stronger, we have a good chance of detecting it. We have some benchmarks going up to about that KD range. So the screening that was designed was to try and figure out what are all the interactions that are possible in the system. That's why we followed it up with the SVR to see how strong the interaction was. But we kind of think about it as a funnel. Our first net with that original screen was to capture every interaction, even the incredibly weak ones. And then the validations, you might figure out things like in your HLA example, like, oh, this one here, maybe the interaction is too weak or something like that is about it. Does that kind of make sense in terms of translating the methodologies?
Yes, yeah. But it means that in the first assay, in the screening assay, there's no way of telling different strength, right?
Absolutely, yes. At least I wouldn't take the data on that interpretation. Some people try and do that, but I think it's a weak correlation. Okay.
And that brings you back then to SPR.
Yeah, exactly. So I think the steps are really critical there to really build a useful network.
Thanks.
Gur, last question.
So I also enjoyed your talk and this unbiased way of quantifying interactions. And along the question that Katarina asked, I mean, considering the strength of interaction, you can also think of nonlinear effects where like many identical receptors will bind to many different or many the same molecules, and the effect will not be just the sum of it, but rather something more. So I understand it's hard to quantify the strength, but could it bias somehow your estimations by having like, non-equal composition of receptors when you estimate the strength?
Yeah, no, absolutely. So to the last part of your question, when we estimate the strength, those are always monomeric soluble proteins. So we're no longer clustering or doing anything like that to, as you just said, avoid some of those biases and confounds. And I think the earlier part of your question, if I understood correctly, is exactly why I cautioned a bit against trying to correlate our initial data in terms of affinity, because those ones, they are multimerized. As you said, that will affect the binding and what you see there to some extent. But yeah, for all the SPR measurements, those were monomeric, gel filtered protein preparations to try and get accurate KDs. Was that answering that, or is there any other parts of that I missed?
No, you answered great. Thank
you. Good. Thank you.
And then maybe the really last question from my side. So you have these protein networks. Are you also trying to look at this from a structural level, like trying, AlphaFold kind of thing?
Yeah. Yes. No, that's a very good question. We, I think I can reveal this. We right now have a collaborator who is working on taking our data set, both this one and some unpublished ones we generated using this really nice method that I developed, that SAVEXIS one, to essentially benchmark, can methods like AlphaFold, can these structural predictions, can docking softwares, how well do they do across these really large benchmarking data sets? I think perhaps even more importantly, a data set that has so many negatives, which usually aren't published, so many things that we measured found not to bound. So I would just say, keep your eyes out for that one. We are working on it. It's a great thing to kind of ask and think about. And I don't have any priors either way. I just handed them the data and we'll see how well they do. Within the next year or so, hopefully that will be coming out.
That's great. Okay, and with that, I think we can close this seminar. It was thanks again to Katarina and Jarrod for these very, very nice talks, and we'll see all of you maybe in the new year, hopefully. Thanks so much again. Bye-bye.
Thanks, everyone. It's a great discussion.